Let's see, there is one more paper what is telling about contamination, and there's additional contamination inside. It's SV40, and it's carcinogenic. And this SV40, it came in this story much later. I heard some explanation how, but let's see what this paper is saying.
Yes, so this is a paper that was published in a journal we saw before, Science, Public Health Policy and the Law. It was published in December by 3 German scientists. It's titled BioNTech RNA-based COVID-19 injections contain large amounts of residual DNA, including an SV40 promoter enhancer sequence. BioNTech, as you might know, co-produced the original COVID vaccine with Pfizer. So it was the Pfizer-BioNTech vaccine. And what this study found, it was a peer-reviewed study, is that the DNA contamination in these vaccines, according to their samples, was 3 to 4 times higher than that 10 nanogram per dose regulatory limit that the FDA and also other international authorities have established. Then, of course, one of the main findings related to that is that looking at the contamination, the vaccines that they looked at were contaminated with simian virus 40, commonly known as SV40. This is the contamination that Kevin McKernan, who we were talking about before, had identified going back to 2003. I spoke to him about this study, and he called it a tour de force on the DNA contamination topic. He wrote on Substack, where he has a blog, that the study's authors did a very good job methodologically. They knocked it out of the park, which is an American expression, that they hit a home run. They did a really, really good job with their study. Jessica Rose, some of your audience may know her. She's an immunologist and biochemist. She's very good at analyzing, statistical data that pertains to, for instance, vaccine safety. She called this the paper of the century. She said it was a very well-written paper. It was a very well-organized study. And this confirmation that it gives us about the DNA contamination, and specifically the SV40 contamination, is significant. Now, why is SV40 contamination significant? This relates again to what we were talking about before, which is the risks that DNA contaminants may pose to the human body. These risks include cancer risks, as we mentioned, and they also include a higher risk of autoimmune diseases, and notably, not just among the vaccinated, but also other people, because the spike proteins that we were talking about before, that the mRNA vaccines also contain, are prone to shedding. So if the vaccine contents are prone to shedding, everything that goes along with that, including these DNA contaminants like SV40, can potentially at least be transferred even to people that are not vaccinated or not up to date, so to speak, with their vaccinations. And this is one, again, of several studies that have come out in recent months, in the past year or so, that have pointed out this DNA contamination. Now, the final and perhaps most important thing of all is that the authors of this paper called for, and this is a quote, an immediate halt of all RNA biologicals. So that includes, of course, mRNA products. So they're saying that the administration of these products should stop. We've seen increasing calls saying the same thing by a lot of organizations. Children's Health Defense has signed on to one of these, and several other organizations of doctors and scientists. But also we've seen, for instance, the Surgeon General of Florida, Joseph Ladapo, come out and say the same thing. We saw recently the report that came from the government of Slovakia, also saying the same thing, that mRNA products are dangerous and should probably be stopped, the administration of them to be stopped. There's other state legislatures in the US that are proposing and debating laws that would place a moratorium on the administration of mRNA vaccines. So there are increasing voices that are not just questioning the safety of mRNA vaccines, but they're also saying, because we have these questions about the safety, even if not everything has been absolutely confirmed yet, there are enough doubts that lead to a very reasonable suggestion that we should at least pause the administration of these products until we have better data.